Hello A2, welcome to your first exciting flipped learning video about localization of brain functioning. So I'm sure you're very excited to get started, so off we go. So first of all, talking about the pink-haired girl, so this is both in the pink-haired girl and the dog book. I've combined the two together. Well, it would be the cat book, wouldn't it, because it's from AS. I've combined the two together. I've also scanned the pages, and they will be underneath on the Moodle page as well. So you can also, if you don't want to listen to this video, you can also just use the information from the textbooks, although you might miss a little bit of the information. Okay, so starting straight away with it, we've got originally there was the view of a holistic view when it came to brain activity, and now it's much more moved towards a localization theory. So the holistic view, this was pre someone called Broca's, pre Wernicke, who were the first people who kind of ever conducted any piece of research. Pre those people, kind of in the early 1800s, there was the view that all of the brain was needed for all of our brain activity. So they've taken that to its fullest conclusion. If you damaged any part of your brain, then you wouldn't be able to complete any activities, which of course we know now. We've seen lots of different examples that's not the case, but it was the belief that the whole of the brain was involved in every single cognitive and any other kind of function within the brain. It all had to be intact to work. Then we had a lovely gentleman called Phineas Gage. You can see a picture of him looking like he'd be lots of fun at parties, and I think we've covered Phineas already. Mr. Gage, so he's packing down some explosive into to blow up some rock on a rail. He was working on the railway, and he forgot to use some pipe or something, so he forgot to protect his the rod in some way. It caught, it sparked, and it flew back through his head, through his eye, and through his brain. After that, he was still able to function as a human being in lots of different ways. He was still alive, he was still very active, he could still speak and engage in everyday life, but his behaviour changed drastically, and that was because of the damage to his frontal lobes. So this was when people started to think, clearly you don't need the whole of your brain to complete every function. Certain parts of the brain impact on certain control certain functions in the brain, and that's when we started to move towards the concept of localization, which is exactly that certain parts of the brain control certain functions within our body, or certain functions that we do. Okay, and we're going to have a look at Broca and Wernicke in a little bit. So just to give you a bit of a taster, give an idea, I'm sure for some of the biology students you've either done this or you'll very soon go over the structure of the brain, but you have two hemispheres, and this is moving on to lateralization. So at the moment we're talking about localization. It's the idea that local areas of the brain, so think local, like little bits of the brain, control certain parts or control certain tasks within the body. Whereas there's hemispheres, which is the concept of lateralization, which we'll look at more at next week. So two halves of the brain. Imagine the brain, cut it down the middle. There's the left hemisphere and the right hemisphere. And you will generally find that the right hemisphere controls the left half of the body, and the left half of the left hemisphere controls the right half of the body. And that's the concept of lateralization that we'll come back to in a little bit. So do bear that in mind, because we do refer to it in this study, in this PowerPoint. However, the focus today is on localization. So small bits of the brain involving certain parts of the brain controlling certain tasks within the body. And then we have the cerebral cortex, which is like the outer part of the brain, and the grey matter, and that makes us talks about the difference between humans and animals. We've developed this grey matter, which is where all the higher cognitive kind of functions come from. Okay, so the reason that I mention that is because in every single hemisphere, in both hemispheres, it is divided up into different lobes. You have the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. And within those lobes, so you have a left frontal lobe and a right frontal lobe, a left parietal lobe and a right parietal lobe, and so on. And then within each one of those, there are certain areas that we have to know about, and that is to do with, they're mentioned on the specification. One of those is the motor area, and there is both a motor area both in the left frontal lobe and in the right frontal lobe, and this controls voluntary movement. And this was a similar time to Phineas Gage, Broca, Wernicke. It was when somebody used probes to stimulate certain parts of the body within frogs, certain parts of the brain within the frog, and certain parts of the body moved. So the motor area is all about voluntary movement, and the way we identified it was localization is we poked a certain part of the frog's brain, we stimulated it, and then we would see a leg move or the other leg move, depending on if it was the other part of the brain. And the way that this part of the part of the brain is structured, the motor area, is in a very logical order. So the part of the brain that controls our thumb movements is also very close to the part, and finger movements is close to the next, close to the part of the brain that controls our hands, and so on. And you will find quite a bit of the brain structure that we talk about is that kind of logical, everything's very close to each other, which would explain why if you have a stroke or some kind of damage, your hand is hurt, your hand is stops functioning, but maybe possibly all your arm does as well, because all of that part of your body that's controlled is all grouped into that area in the brain. And it says lateralization at the end because there is a motor area in the left and a motor area in the right. The left motor area in the left part of the brain controls the right side of the body, and the right side of the body is controlled by the left motor area. Again, so both sides of the brain control the other side of the body. So it's both localization and lateralization because it's a local part of the brain. It's in a certain area, the frontal lobe, but also it's on both sides, and the opposite then controls opposite sides of the body. Then they've got the somatosensory area, which is in the parietal lobe, and again this is it is controlled through lateralization. So you do have a left and a right. You have a somatosensory area in both the left and the right hemispheres in the same place, and this takes in the information of sensation from the skin. And again, this is structured quite logically, so a very large part of the somatosensory area in the brain is given up to the kind of the most where the most sensory information comes from in the body, so fingertips, things like that. And then this also links into another concept that we're going to look at later on, which is plasticity. So not only are the biggest areas of the sort of the most significant areas of the body that take in the most sensory information, the most dominant, the largest within the somatosensory area, the area also experiences plasticity. So this is the idea that the brain is shaped by experience. So when we're born, we're not just born with a brain exactly as it's going to be. It's shaped by different experiences, or it doesn't stop growing at a certain age. So Braille readers, for example, have a very, very strong, a very enlarged somatosensory area within the brain because it relies very carefully on very sort of sensing physical, very small physical differences when you're reading about Braille. So they rely on that area a lot more than probably people who don't read Braille do. So it does respond, and that could be somebody who started reading Braille when they were 20 compared to someone who's always had to read Braille. The brain grows, but that's more to do with plasticity. Visual area, which is in the occipital lobe, and this is all about processing imagery. So it processes the visual imagery that is presented in our brain, and again this is to do with lateralization, and we will look at Sperry's split brain research when we talk more about localization, which does look at the idea that information seen in the right visual field is processed by the left hemisphere, and then information coming from the left visual field is processed by the right visual hemisphere as well. And then we have the auditory areas, which is in the temporal lobe, and this analyzes acoustic information, so information that we receive through our ears. And again, this is structured with the idea of lateralization. So if you hear something in the left, it's processed by the right, and if you hear something in your right ear, it's processed by the left brain as well. I think I just repeated myself in a different order there, but you get the idea. And then there are the language centers. This is really important. This one does. Language centers are not lateralized. They are, and we'll look at the next slide is a bit more of an extension task. So if you're thinking, well, all of this information is more than enough, then you don't need to go into this any further. So what you need to know, if you're kind of keeping it basic for now, ready to build later on, is that the language centers, there is no lateralization, and they are almost always in the left hemisphere. And we have two types of language centers. We have Broca's area and we have Wernicke's area. Broca's area is in the left frontal lobe and is all about speech production. So this part of the brain helps us to produce speech. So I'm sure you're all thrilled that my Broca's area is clearly working very effectively at the moment, and I'm able to produce fluent and hopefully speech that's fairly understandable as well. If that part of the brain is damaged, then this person experiences expressive or Broca's aphasia. And what that means is that they struggle to express themselves essentially because the brain is damaged. So that tells it is speech production. And there was a gentleman called Tan who Broca studied, who was called Tan because the only word he could say is Tan. When poor old Tan died, they conducted an autopsy and they looked at his brain, and found that this part, the left frontal lobes, that part of Broca's area was damaged. So Broca kind of went, yep, I'll name that then. And it was all about the fact that they knew it was focused on speech production. The two YouTube links are really, really interesting. If you just Google Broca's aphasia, I think they'll both come up as well. They're about an 18-year-old called Sarah Scott who suffered a stroke. It's very interesting to watch her try and explain that experience. And then if you're when she's 18, and now she's 25, 26, and there's an update on how she's doing as well, which also links into the idea of plasticity. So she really, really struggled. She experienced a stroke and she struggled to express herself, or experienced Broca's aphasia. And you can see that in the first video, and then gradually by the next video you can see that her speech has improved, and that's potentially because of plasticity. So other areas in her brain in lots of different manners, different ways, have taken over that function. It's quite interesting. And then Wernicke's area, so another language centre that's in the left temporal lobe that isn't lateralised, so there's no right a Wernicke's area in the right temporal lobe. Which is about speech comprehension, and that is about understanding speech, and being able to just understand it. Sorry, clearly my Broca's area is damaged there a little bit. So speech comprehension, and that's about receptive. It's struggling to receive and understand information, or Wernicke's aphasia. If I were you in the exam, I'd encourage you both to use expressive and receptive aphasia, purely because it just shows that you know some more information, some other words in there as well. Okay. And then this is the extension. So if you're thinking, yeah, whatever you've just said was more than enough, I don't think I can cope with any more, then you can leave it. And I'll be honest, a lot of this information I didn't know, having previously taught the A2s, and they all passed absolutely fine. So it's not essential, but maybe I know we've got quite a few biologists or people who are wanting to access the higher information, the higher grades. You can have a look at this information, and I'd also encourage you to go away, look in the cat book, look in the green-haired girl book, both of which are scanned underneath in the Moodle, and get some more ideas. So the motor, just a little bit of additional information, the motor cortex is in the frontal lobe, in the precentral gyrus, and the somatosensory area is in the parietal lobe and is in the postcentral gyrus. So you can have a look at what those terms mean and get a bit of an idea of the structure. And then there is the visual area, which is in the occipital lobe, and possibly you probably remember this from GCSE and may probably remember it more clearly than I do, but you could talk about the process of information, light waves coming into the retina and being processed by the photoreceptors, so rods and cones. That information is travels down the optic nerve into the thalamus, which is part of the brain, which then relays the information to the visual cortex, and the visual cortex processes it and understands it in more detail. Tons of information on YouTube, tons of information on the internet, so have a bit more of a look at that. And then the auditory centre, you've got it in the temporal lobe, and the sound waves come into the cochlea and are transferred into nerve impulses, run down the auditory nerve and into the auditory cortex. And the brainstem and the thalamus is also involved in the same way as it is in the occipital lobe, with processing and understanding that information and what it means. So not just nerve impulses, what's the information that's being provided. Then Broca's area is in the posterior inferior frontal gyrus. If you'd like to remember that, obviously can impress people at parties, and there are two areas to it. So there has been research to suggest that not only is Broca's area involved in speech, but also another area of it which was active when people weren't speaking was cognitive processing. So they did a brain scan, and when people weren't speaking but Broca's area was still lit up, and they felt that was because they were doing a cognitive processing task. And then you've got Brodmann's area 44 and 45, which is where, so the area 44 is more motor aspects of the speech. So Broca's area not only linked to speech production is not only to do with forming the words, as in knowing what to say, but it's also about physically how to form them. So, and that's very, very close to the motor area, motor cortex, and the area that involves like control of our mouth, so chewing, things like that. Whereas this area is probably more about the motor aspects, controlling our mouths to move our tongues and all that kind of stuff for speech. And then you've got area 45, which is more about semantic decision tasks, so kind of understanding and processing, categorizing words and thinking of new words. So for example, generating a verb associated with a noun. So they kind of understand meaning and and what words to generate from that. And then vernix area is in the posterior section of the superior temporal gyrus. So again, another very impressive sounding term. Oh, I should add, Area 45 does have, it's quite controversial apparently. So from the very limited reading I've done, it's very controversial, the kind of tasks that it's involved in. And then this is the bit that I made reference to briefly earlier about the dominant hemispheres. So for anybody who's trying to keep it simple for now, and I don't blame you, Broca's area and vernix area are in the left hemisphere. However, you can remember that. However, it is actually in the person's dominant hemisphere. Now, the majority of the time, that is the left hemisphere, because it's the right side of the body that's dominant. Think, obviously, there's more people who are right-handed than left-handed when they write. However, the dominant hemisphere is left in only 95% of right-handed individuals. So the right hemisphere is dominant 5% of the time, and then in left-handed individuals, bearing in mind lateralization. So if you're left-handed, it's the right side of the right hemisphere that's controlling the left hand. So actually it would make more sense that more of the left-handed people, it had the dominant hemisphere was the right. So you can find Broca's area and vernix area in the right hemisphere. However, it's rare, and if you weren't quite sure how to explain that or didn't have time, last year I taught everyone that it was in the left hemisphere and that we didn't discuss it any further than that. But you can add that level of discussion, you know, if you're running out of things to say and you've got loads of time left in the exam, which I know happens to everybody. So, and then there's just a visual image, and I'd encourage you to have a look at some different imagery. Word of warning, every single person will be expected, potentially, definitely in my classes and likely in the exam, to label all the different areas of the brain. So you need to know about the frontal, occipital, temporal and parietal lobes. You need to know where they are, and then you need to be able to tell me where the motor cortex, the somatosensory cortex, the visual cortex and the auditory cortex are. So there'll be arrows pointing to the different parts of the brain, but you've got to remember which arrow links with which thing, and we'll work on that in class, but that's definitely something you want to think about at home as well. Right, evaluation. So we're just going to do some brief notes on evaluations, talk to them briefly today. The way you structure evaluations will make the difference in the bigger mark questions from sort of the lower grades to the higher grades. A 16-mark question, you could get a 16-mark question on any of the topics we've talked about, 6 marks evaluation and 6 marks description and 10 marks of evaluation. I have included 4 evaluation points, and there are loads more in the book, although some of them are a bit naff. The number you will need that will easily, if you do them properly, will allow you to access the higher grades is 3. I would rather you focus on quality over quantity, which I know we've said throughout the whole year. You do not need more than 3. I've included 4 in case you think, yeah, I don't get that one. So you've got an alternative one, and there are loads in the books as well that you could look at. If you don't like any of these, do not overburden yourself with more than 3 though. Supporting research. There are 2 well, there's loads of supporting research, but there's 2 pieces of supporting research we are going to look at. You've got Doherty in 2002, who looked at patients with OCD who had cingulotomies, and that means there's a certain part of the brain was lesioned, and lesion just means cut or sort of essentially destroyed, damaged in some way. So of these OCD patients, they went in, they had the neurosurgery, and let me just check my notes. What were the numbers? They found that a third of the patients who were previously experiencing OCD had felt there was a reduction in their symptoms, and 14% had a partial response as well. So that could be viewed as fairly successful, considering these neurosurgery for mental health conditions is generally quite rare. We only do it if the patient is really, really desperate for some kind of relief and hasn't, and no kind of more first line. So the first things we would consider have worked. So what you would need to do though is you need to structure that in a P point with Doherty as your example. So your point would be that there is supporting evidence that comes from research into neurosurgery. So neuro just means brain, so brain surgery, but obviously neuro makes you sound all fancy and clever A-level psychology studenty. and then you would give your example of Doherty, and the key thing you want, they're not really that bothered about the procedure, they are more interested in the findings. And then you would say, and this supports the process, the concept of localization because, and then you would explain why. So quite clearly, if it supports the idea that there are mental health can be localized to certain parts of the brain, so clearly certain parts of the brain do control certain behaviors, suggesting that localization theory has validity. So you want to really, really make sure this year is the year to improve your evaluation points. And then you've got Peterson. So another piece of research there is also supporting research that comes from brain scans. For example, Peterson, who found that when participants were completing a listening task, he found that Wernicke's area was activated in the brain, whereas when they were completing a reading task, Broca's area was activated in the brain. And then this would be the bit that gets you the higher marks. So what does that suggest? Well, clearly it suggests Wernicke's area is all about comprehension. So if Wernicke's area is active when people are listening, then they're obviously trying to understand and appreciate and comprehend speech. And then Broca is all about is activated when we're trying to produce speech. So when they were reading this speech information, so reading it out loud, then Broca's area was needed so that they could produce that speech, and that supports it. So then again, that supports the idea of localization, that certain parts of the brain are in control of certain tasks, increasing the validity of the localization theory. And then finally, opposing research. So you've got equipotentiality theory, and as you can tell, I'm fluent in this theory. I am aware of the theory, and I'm comfortable with the concepts of it. I just cannot pronounce it, so please don't ask me in class. And this is the idea that actually they're combining the two ideas of both holistic theory and localization. So there are certain more basic tasks which are controlled by certain parts of the brain. However, when it becomes more complex kind of cognitive tasks that require a lot of thought process, then the whole of the brain becomes involved with that. So, and there's a couple of different things to support that, and you can choose what you talk about the more that you see. So this is supported by the idea of plasticity, that people who experience brain damage and therefore certain cognitive functions or functions of any sort are stopped for a while. However, the other parts of the brain can take over, for example, speech or movement, anything like that, or comprehension. So the brain is very elastic and plastic. So you can so it can take over different functions. Just because, say, for example, Broca's area was damaged altogether, it wouldn't mean that the person could never speak again. It might, but it's not guaranteed. And then we also have Lashley and the rats in the mazes. And he removed different or lesioned different parts of the area in the rats' brains, varying between 10% and 50% while they were learning the maze. And he found that no particular part of the brain stopped the rats. So there's no particular part of the brain, the localization of the brain, that was all about maze learning. He didn't find there was one particular bit that if you damaged it, the rats couldn't learn how to run a maze. He felt that it was all over holistic. It was the whole of the brain that was getting involved, trying to process the task, which obviously reduces the validity of the localization theory, because you would expect that there would be a certain part of the brain that could be damaged, and therefore the rats couldn't run the maze anymore, calling into question the conclusions. You could also talk about Gabby Gifford, who we do look at in plasticity as well. So she was a lady. So there'd be evidence that opposes the localization theory in the form of case studies. For example, Gabby Gifford, she was a lady who was shot and she couldn't do certain things, and if you look in the video you'll see there are quite a few things that she wasn't able activities she wasn't able to complete. However, 4, 5, 6 years later, she is able to, her other parts of her brain have taken over the damaged areas, and she's still able to perform those tasks. And of course that calls into question the idea of localization, because once a certain part of the brain is damaged, it's the only part of the brain that can control that task. Although you could argue that that's not necessarily the case. Again, with all of these, you are able to add research methods, evaluation points. So what's the problem with Doherty's? I think it was, let me just check, how many 44 OCD patients, so they're an abnormal kind of clinical group, which suggests that they're quite unusual. And bearing in mind the way that we use the term abnormal in psychology isn't the way that you'd use it as an insult, it's just a description, so as you all know. So abnormal a clinical group, very small, 44 isn't particularly representative. Brain scans, so we will look later on, we will look at problems with brain scans, and you could talk about the fact that they only provide quantitative data, or they do have their limitations, and we'll come back to that one, unless you'd like to read ahead to ways of studying the brain, and then you can already do that. Plasticity, particularly Lashley with the rats, but of course you've got problems with transferring information from animals to humans, and I'm sure you can remember the term escapes me now at the moment, the one we use all the time. Anyway, I'm sure you can remember that about extrapolation, taking information from animal studies and applying them to humans. Rats' brains are far, far, far more simple than human brains. And then Gabby Gifford, of course, is a case study, so there's issues with population validity there. She's a very unusual and unique case, and really it would need to be replicated before we could be confident of the reliability or the validity of the study, because there are lots of different factors. And that is that. So that's your first soiree into a little bit of biopsychology. Hopefully you're not all terrified now. If you do have any questions, let me know. Otherwise, can you just make some notes on your books and make sure that you bring them for next week? Thank you very much.